Hello everyone, my name is Cara Goldman, and today I'm with my partner, Sophie Martin, and today we'll be presenting the results of our microbiology project, which focused on how ultraviolet radiation affects bacterial survival when the bacteria have been treated with different levels of sunscreen. We tested SPF 15, SPF 30, and SPF 50, and then exposed the bacteria to UV light for different time intervals to see how long it took for them to die. The purpose of this experiment was to understand how sunscreen acts as a protective barrier, not just for humans, but also for bacterial cells, and to explore how different bacteria respond to UV stress. Throughout this presentation, I'll walk you through the background science, our hypothesis, detailed methods, and the results we observed, and what these findings mean. UV light is a form of radiation that damages DNA by forming thymine dimers, basically creating abnormal bonds between thymine bases in the DNA strand. This disrupts DNA replication and transcription, meaning the cell cannot divide properly or repair itself. If too many dimers form, the cell eventually dies. Because of this, UV light is commonly used in laboratory sterilization. In our experiment, we use this DNA-damaging property of UV to test how sunscreen may help reduce its effect on bacteria. SPF, or sun protection factor, measures how well a sunscreen can block UV radiation. For example, SPF 15 filters out around 93% of UVB rays. SPF 30 filters out about 97%, and SPF 50 filters out approximately 98%. These numbers might seem close, but every extra percent represents a large reduction in the amount of UV reaching the cells underneath. Sunscreen protects human skin by absorbing or reflecting UV radiation. Our project aimed to determine whether sunscreen could also protect bacteria in the same way. Our hypothesis was that sunscreen would protect bacteria from UV radiation, and that higher SPF would provide stronger protection. All plates received sunscreen, but only some were exposed to UV. Each SPF level included a control plate and a UV exposed plate. We predicted that for each SPF level, the control would have the highest survival, and that among exposed plates, SPF 50 would show the most survival, while SPF 15 would show the least. To start the experiment, we prepared serial dilutions of both Staphylococcus epidermidis and Bacillus subtilis at 1/10, 1/100, and 1/1000th serial dilution. We plated each dilution to determine which concentration would give us a reliable number of colony-forming units. In microbiology, 200 to 300 colonies is considered the ideal range. Below 200 is too sparse to be reliable, and above 300 becomes too crowded to count accurately. When we evaluated our test plates, the 1/10th dilution consistently produced around 300 colonies, which was perfect. Therefore, the 1/10th dilution was selected and used for all plates in the experiment. Now, after choosing our dilution, we inoculated the plates using sterile technique. Each plate received the same volume of the 1/10 bacterial dilution. We then used sterile spreaders to evenly distribute the bacteria across the agar, which is important for ensuring consistent colony formation. Now, during inoculation, we labeled every plate with the species, SPF level, UV exposure time, and whether it was a control or an exposed plate. Inoculation occurred before sunscreen application so that all plates began with the exact same bacterial load. Once the plates were inoculated, we applied the sunscreen. Every plate in this experiment received sunscreen so that the only variable between control and exposed plates was UV light. To keep the experiment standardized, we applied the sunscreen using the same number of sprays and the same distance from the plates. Now, this ensured that all the plates received an even layer of coverage. After application, we allowed the sunscreen to dry completely so it would form a consistent protective barrier. For UV exposure, we used a UV lamp positioned in a fixed height above the plates to keep the intensity consistent. We exposed the plates to a range of times: 30 seconds, 45 seconds, 60 seconds, 3 minutes, 4 minutes, and 10 minutes for both species. Only the exposed plates were placed under the lamp. Control plates, although treated with the sunscreen, were kept covered and away from the UV light. This ensured that the only difference between each pair of plates was whether they received UV radiation. Now, in this slide, we are looking at a direct side-by-side comparison of Staphylococcus epidermidis and Bacillus subtilis after being exposed to UV light for 30 seconds, 45 seconds, and 60 seconds. This is one of the clearest ways to visualize the biological differences between these two organisms. On the left, you'll see the staph plates. Even at 30 seconds of UV exposure, there is already a noticeable reduction in colonies. At 45 and 60 seconds, the colony numbers continue to drop significantly. Now, this shows that Staphylococcus epidermidis is highly sensitive to UV light, and even short exposures can cause rapid DNA damage and bacterial death. On the right, the Bacillus plates tell a completely different story. At the exact same exposure times, 30 seconds, 45 seconds, and 60 seconds, Bacillus subtilis shows much heavier growth. The colony numbers remain high, and the plates look much more filled in compared to the staph plates. This stronger survival is due to the fact that Bacillus subtilis can form endospores, which are extremely resistant structures designed to protect the bacterial DNA from harsh environmental conditions like heat, chemicals, and UV radiation. Even when all the cells are not sporulated, Bacillus still has significantly more natural resistance than staph. Now, this comparison shows that the species of bacteria is just as important as the SPF level when predicting UV survival. Staph dies quickly under UV because it is a non-spore forming bacteria, while Bacillus maintains high survival because of its endospore forming ability. These differences become even more pronounced as UV exposure times increase. For Staphylococcus epidermidis, the graph and plates both show a very clear pattern. The lower the SPF, the faster the bacteria die under UV exposure. SPF 15 plates lose survival quickly, even at shorter exposures. SPF 30 provides moderate protection, but eventually decline as exposure time increases. Now, SPF 50 consistently maintains the highest survival levels. These results reinforce the idea that sunscreen acts as a protective layer that slows down the rate of UV-induced death. Now, in Bacillus subtilis, SPF still plays a role, but the species' natural UV resistance dominates the survival pattern. Even at 10 minutes, Bacillus plates have visible growth, especially with the SPF 30 and SPF 50. The SPF trend remains the same. 50 performs best, then 30, then 15. But the survival rates are significantly higher than staph. This again shows how biological structure influences UV resistance. At intermediate exposure times, the SPF differences become even more pronounced. Staph experiences dramatic death between 60 seconds and 4 minutes, while Bacillus maintains strong survival. SPF 50 continues to outperform the lower SPFs in both species. This middle time points are important because they show the beginning of major separation between SPF levels, indicating that the protective effect of sunscreen depends not only on SPF, but also the length of UV exposure. Now, the 10-minute exposure showed the strongest effects. Staph was nearly wiped out across all SPF levels, although SPF 50 still maintains slightly higher survival than SPF 30 and 15. Bacillus, however, still showed visible survival even after 10 minutes. This final time point strongly reinforces the species differences and confirms that SPF reduces UV damage but cannot completely stop it. Our discussion centers on two main points: the protective role of SPF and the different biological features of the bacteria. So higher SPF levels consistently slowed bacterial death under UV radiation. This effect was observed in both species, but the degree of protection varied. Bacillus, due to its ability to form endospores, naturally resisted UV damage much better than staph. These findings support our hypothesis and align with known microbiological principles. The data is strong, consistent, and biologically meaningful. In conclusion, the UV radiation is highly effective at killing bacteria by damaging their DNA, but sunscreen can significantly slow this process. Higher SPF levels offer stronger protection, with SPF 50 showing the best results. However, the biological characteristics of each species plays a major role. Bacillus subtilis consistently survived better than Staphylococcus epidermidis due to its ability to form UV-resistant endospores. Overall, our findings strongly support the hypothesis and demonstrate how environmental protections like sunscreen can influence microbial survival. Thank you for listening, and I'd like to acknowledge my instructor and classmates for their help throughout the project.